Hi, everybody. I'm here today to talk about a technology that we developed in the Boyer lab to fill a crucial need in the pharmaceutical industry. So I think we can all appreciate that we get sick, we take drugs, and those drugs have to affect a certain population of cells in our body. And in the pharmaceutical industry, in the pursuit of developing treatments for new diseases as they emerge and old diseases more effectively, pharma companies are turning toward more complex drugs. But these drugs still have to get into the cells to do what they need to do. And with more complex drugs comes more complex drug delivery, particularly at the cell level. So we saw a need for a technology to address the study of these kinds of drugs. And there are a lot of barriers associated with a drug getting to where it needs to go to do its therapeutic work, from navigating the bloodstream to avoiding the liver. But what I want to highlight is that in order for a drug to be effective, it has to not only get inside of a cell, but it has to get to a specific place in that cell where it can do its therapeutic work. And in the pharmaceutical industry, it's really difficult to study that process. And so we saw a need for a tool to sort of interrogate this little cellular highway. So for studying cells in the lab, typically what we're going to do is take a cell type of interest and isolate it. We'll culture these cells in a multi-well plate where we can run an experiment on them. And then after that experiment, we'll take our cells to a microscope where we can image the cells. And the end result of that might look something like this. What you're seeing is two cells, one on top of the other, and you're looking at a structural protein, which is sort of like the skeleton of the cell. And that's all well and good, but if we really want to interrogate those cellular highways that I talked about, we need to increase the resolution of our images. And the question becomes, how do we get that increased resolution while using the same high-throughput microscopes that pharmaceutical companies already use? So the answer to that is a technology that came out of the Boyden lab in 2015 called expansion microscopy. To understand how expansion microscopy works, imagine that I have a deflated balloon and I write some text on it, the word cell in this case. As I inflate that balloon, the text on the surface is going to expand with the surface of the balloon. And you're going to get a bigger word that's easier to read. And we could do something sort of similar in cells by taking them and applying a hydrogel solution and polymerizing that solution on the cells to effectively embed the cells in a polymer matrix, which we can then expand. And by virtue of that expansion, we can get a larger cell that, just like the word on our balloon, is sort of easier to image. And we can take a look at what this looks like on a slice of mouse brain tissue. And you can see as it physically gets larger, you can start to pick apart some of those smaller details. So that's cool. We have the technology, but in order to adapt that to screening, like a drug screen, we need to streamline the implementation of expansion microscopy. So where typical implementations involve sort of sample-by-sample preparation all the way from the cell culture to the image acquisition, we developed a technology that would allow us to carry out this process dozens of times in parallel in a 96-well plate, which really opens up the possibility for using super resolution microscopy for a screen. So this is the technology that we came up with. It's a plastic well plate insert that we filed a provisional patent on earlier this year. It's inexpensive to manufacture. It's easy to use. The way that it works sort of centers on this small feature at the tips of these posts that go into the wells of the well plate. The tips of these posts will collect tiny volumes of the hydrogel solution that we use for the process of expansion microscopy. And those droplets can then be deposited into the wells of the well plate where our cells are, polymerized using ultraviolet light, and expanded. So at this point, we can take our cells of interest, expose them to drugs that pharmaceutical companies are interested in studying, and we can start to really pick apart the compartments in this sort of cellular highway that I've been talking about, where a drug might get caught up or is getting trafficked through. And we can start to see more detail in these compartments, and through the use of these high-throughput microscopes that pharmaceutical companies use, we can take these images by the thousands to start to inform drug discovery or drug design in a new way. So we have a small team of Multilogics of past and present members of the Boyer lab. We're looking to expand our reach a little bit. And so we have this technology, and I talked about one sort of use case in the pharma space. But at the end of the day, what we have is a tool for high-throughput, super-resolution imaging. And so you can imagine the potential application space of that tool is pretty expansive. So we're looking for scientists who can take our tool and use it to enable their science in a new way. If you have any ideas, I'll be around. Let me know. And thank you for your time.